Hello students, welcome to this particular module. In this module, we are going to discuss about Selaginella, its morphology, its anatomical details and also about its reproduction. Before that, we need to know the systemic position of Selaginella. It belongs to the division Lycophyta, class Lugolopsida, order Selaginellae and family Selaginellaceae. It has a wide range of occurrence. Selaginella comprises of almost 700 species and is known as spike moss or club moss. They usually prefer to grow in moist and shady hill areas. They grow in areas which is less illuminated and is a little bit humid, especially in soil floor. It is found growing abundantly in the tropical rainforest. Two of its type, one xerophytic and one epiphytic, are found. Epiphytes are those which are growing on tree trunk and xerophytes grows in areas where there is a stress of water or in drought conditions. These xerophytic plants are also known as restoration plant. That is, they have a capability to regenerate itself after the drought period is over. Here I have given the pictures of the said plants. Here is the normal Selaginella that I have talked that they grow in tropical forest. Here is the epiphyte and this is a xerophyte. Now habit. They form a delicate green mossy cushions. Some are vine-like and the others are creeping axes with a leafy branch system. There are certain salient features of this Selaginella. Firstly, it comprises of microphyllous leaves, that is, the leaves are very small. The sporophyll are arranged in a strobilus and is placed terminally. A special structure called rhizophore are present. It is heterosporous, that is, two different type of spores are produced. The gametophyte is dioecious and the anthrozoids produced are biciliate. The two important features of this genus is heterospory and the reduction in the gametophyte. The heterospory, as I told, is the production of two different type of spores in two different sporangia. I have given a figure for that where you can see this microsporangium and megasporangium each bearing the microspores and the megaspores. The second one, the extreme reduction of the gametophyte. The male gametophyte is reduced just to antheridium and the female gametophyte produces archegonia with some storage tissues. This genus can be subdivided into two subgenus, the homophylum and the heterophyllum. The difference between these two is basically in their leaf structure and also in the stem. Apart from being monostelic, this homophylum is monostelic, whereas this heterophyllum is monostelic but contains a ribbon-shaped stele. This particular plant body is differentiated into the root portion, the rhizophore, the stem, the leaves. And at the apical portion of the leaf structure, we find that it is terminated into a strobilus. We will be discussing each portion separately. The first one is the stem. The stem is profusely branched, delicate and evergreen. The branching type is monopodial type. Here I have given a particular diagram where you can see that how the monopodial branching system takes place. The growing apex of the stem consists of either meristematic tissue or a single apical cell. Here, the homophylum and heterophyllum can be differentiated. In case of heterophyllum, the particular stem is a prostrate type, but in case of homophylum, it is erect. So, homophylum has erect and cylindrical stem. Heterophyllum has a prostrate stem with stout erect branches. Now we will be discussing about its internal features. First, internally, The stem can be differentiated into the epidermis, forming the outermost layer, the cortex forming the middle layer, and the stele, which is centrally located. So if we consider this picture, we can easily find the outermost epidermal layer, the middle cortex layer, and at the central is placed stele. The outermost layer is one layer thick, the cells are cutinized, and there is an absence of stomata. The middle cortex is differentiated into the outer cortex and the inner cortex. The outer cortex is thick-walled and is sclerenchymatous, and the inner cortex in parenchymatous. There is an absence of intercellular spaces. Here in the diagram, we can see that this is the cortical portion, the sclerenchymatous portion, the parenchymatous portion. Now, this outer cortex is attached to the central stellar portion with the help of some elongated cell which is endodermal in origin, known as trabeculae. This inner portion consists of the vascular bundles, or the stele. This stele comprises of the pericycle, the xylem, and the phloem. Here we see that there is no pith, so it is protostelic in nature. Before discussing in details, I want to say that in Selaginella, in some species, a polystelic condition is also found where there is a presence of two different types of steles. So, each stele is comprising of the pericycle, phloem and xylem. Pericycle is thin parenchymatous cell. Phloem consists of sieve cell, phloem parenchyma, but there is no companion cell. The xylem is exarch and one or two protoxylem groups are also found. It comprises of only tracheids. Now we come to the root. The root of the young sporophyte is ephemeral, but as it matures, it becomes adventitious. Here also, the outermost layer is the epidermis, and above the epidermis, a cuticle is present. The specific characteristic of this internal structure of root is the presence of these root hairs, and these root hairs are epidermal in origin. After the epidermis lies a hypodermis formed of sclerenchymatous cell. There is a cortex formed of parenchyma, and internally we can see there is a endodermis, a pericycle, and a layer of phloem. And at the middle there is a xylem, that is xylem is being surrounded by the phloem. While beginning, I told there is a specific structure found in this genus. This is known as the rhizophore. The rhizophore is a cylindrical structure that originates from the stem, and at the lower portion near the soil, some roots are being formed. Here, it also has a single layered epidermis followed by two cortex, one of sclerenchymatous cells and the other of the parenchymatous cell. The rest are almost similar as we have seen in the root. Now there are lots of theories that have been proposed regarding the nature of this rhizophore. Since we all can see, these rhizophores are positively geotropic, that is, it is growing down, and no leaf structure are seen growing from that particular structure. As a result, we can say it is a root. Again, there is no presence of any root hair, so we can say it is again can be a stem. Regarding this, three theories have been proposed. One is the capless root hypothesis. Second, the leafless shoot hypothesis. Here they have added that it arises from the angle meristem present at branching. But the most accepted one is the sui generis hypothesis, that it is neither a root nor a stem but a new structure that is being formed in this particular genus. Now we come to the leaves. The leaves are microphilous, sessile and simple. So if we see that we told before that the homophylum and heterophyllum can be differentiated on the basis of these leaves. What is that? In case of homophylum, the leaves are isophilous, that all of them are similar. But in case of heterophyllum, the leaves are of two types. One is the smaller one and the other is the larger one. Now, we go into the internal structure of the leaf. What we see here? We see that there is a thick epidermis. Now, this epidermis is two-layered. By two-layered I mean one is in the upper portion and the one is in the lower portion. The middle portion is being occupied by the mesophyll tissues. We see that in many plants, the mesophyll tissues is again divided by pellucid parenchyma and spongy parenchyma. In Selaginella, there is a variation. In some, the mesophyll tissues is not differentiated, but in many, we see that it is differentiated. Stomata is present in the abaxial surface, that is on the lower surface. And in this mesophyll tissues, some air cavities are seen. There is a peculiarity regarding the chloroplast present here. The chloroplast is cup-shaped. See, I have given a picture of the chloroplast here. This is a cup-shaped structure and inside this, some cells are present which looks like pyrenoids. Then we come to another substructure, that is the ligule. The ligule is a small structure present at the base of the leaf. This is the leaf at the base of the leaf. This ligule is present, which is a tongue-shaped structure. Internally, this ligule can be differentiated into two portions. One is the glossopodium and the other is the body. The glossopodium is present at the lower portion and it is surrounded by a glossopodial sheath. The upper portion, that is the body of the ligule, is formed of various types of cells, that is some are smaller and some are larger. The proper function of ligule is not yet known. Maybe it has some protective function or it is used to keep the sporangium wet. Now we will be coming to the third portion, that is the reproduction of this particular genus. In this particular genus, three types of reproduction are seen. One is the vegetative reproduction, one is the sexual reproduction and the third one is the sexual reproduction. In case of vegetative reproduction, it takes place by fragmentation, by resting birds and by tubers. Asexually, it reproduces with the help of megaspores or microspores, and sexual reproduction takes place by the formation of male gamete and the female gamete. So first we will be discussing about the vegetative structure, fragmentation. Sometimes some adventitious branches grows from the stem. These adventitious branches can detach from the parent plant and can give rise to a new plant. The tubers are aerial and are present in this root structure. See, here it's the particular tuber. This tuber can again under favorable conditions form a new plant. Lastly, we will be talking about the resting birds. Here we see the resting birds. It develops at the apical end of the aerial branches. The leaf regions are closely arranged and overlap the growing points. When the conditions are favorable, these resting birds can grow into a new plant, so it can withstand the unfavorable conditions. After this, we will be discussing about the asexual reproduction. So as I mentioned earlier, asexual reproduction takes place with the help of spores. Now, the spores are present in a structure called strobilus. So this strobilus is a reproductive structure formed by the aggregation of ligulate sporophylls at the apex of the branch of the stem. The growth, sometimes in some species, the growth of the stem continues beyond the strobilus, and such a condition is called selago. Selaginella is heterosporous. That means it will be producing microspores and megaspores. These microspores will be produced inside the microsporangium, which will form the male component for this sexual reproduction. And the megaspores will be formed inside the megasporangium, which forms the female gametophyte. Actually, this difference in the spores lies in their sizes. The microspores are very small in size and are produced in larger number, whereas the megaspores are produced in smaller numbers and are very large in size. Here it is so. Now, what is this position of the sporangium? The sporangium are present in the sporophylls. Sporophylls are the leaves. So, both the leaves for microsporophylls and megasporophylls are the same. In case of microsporophyll holds the microsporangium and the megasporophyll holds the megasporangium. Now, we will be discussing about the development of the sporangium. This sporangial development is usually eusporangia type. So, it will be formed of a group of sporangial initials. The sporangial initials are usually the superficial cells. These are the sporangial initials. Now, there will be a divisions which will form an outer cell and an inner cell. The outer cell will form the jacket initials, whereas the lower cell will form the archisporial initial. The jacket initials, there will be further division to form the jacket layer. The archisporial cell will form the upper cell and the lower cell. The lower cell will form the sporogenous tissues, but the upper cell will form the tapetal layer. This tapetal layer is just a nutritive layer. The lower portion of the sporogenous tissue will form the stalk. So, ultimately, a sporangium will be formed having a two-layer thick jacket, a single tapetal layer, and inside it there will be the sporogenous tissues. During this stage, the microspores and megaspores are not formed. Now, the megaspore mother cell will be formed. This megaspore mother cell will divide mutatically to form the microspores. And all the microspores will be functional. But in case of megaspores, all the megaspores will not be functional. Only the functional megaspore will form the megagametophyte. Now, here is the microspores and the megaspores. You can see very well that both are, one is very smaller as compared to the other. There is an outer layer called the exine and an inner layer called the intine. In case of megaspores, there is a third layer known as the mesosome layer is also present, known as this mesospore. Actually, this megaspore is present in form of a tetrad, and when it is separated, this single megaspore is there. Now, we will be talking about the development of the male gametophyte. So, male gametophyte will be formed from the microspores. This or the formation of male gametophyte or female gametophyte takes place within the microsporangium or megasporangium, that is within the spores this development takes place. So, this is the microspores. There will be a first division. This division will be unequal, which will form a small prothalial cell and a large antheridial initial. In this development of gametophyte, we will see that there is a lot of development of this jacket cell. See, there will be a first division in which the first jacket cell will be formed. Second layer of jacket cell will be formed. Again, there will be division. Third layer of jacket cell will be formed, and ultimately the fourth jacket cell will be formed. And in each division, two jacket cells are being formed. Here in this diagram, you can see that how this particular division is taking place for the formation of the fourth jacket cell. And ultimately, there will be another division in which the androgonial cells will be formed. These androgonial cells will form the sperm mother cell. Now this sperm mother cell is being formed. Now a particular situation is attained where it is a 13-celled structure. We have 8 jacket cells, 4 androgonial cells and the 1 prothallal cell which was not further divided. So during this 13-cell structure, this particular structure will get detached and further development will take place. The sperm mother cell will now metamorphose to form the anthrozoids. These anthrozoids will be a biflagellated structure and thus the sperm, that is the male gamete, will be produced. Now we'll be talking about the development of the female gametophyte. The development of female gametophyte occurred with the formation of a vacuole. A vacuole is formed and at the outer layer there will be the nuclear divisions, that is the cytoplasm will undergo the nuclear division and ultimately at the side there will be this divided structure and at the middle a vacuole will be formed. So this will be the ultimate structure in which 3-ridge structure and from the apical portion some apical cells will be produced below the ridge. Now at this stage this particular gametophyte will get detached when it will get a proper substratum. Now it will exert some pressure on the wall will break and the rhizoids will get exposed from there. So here you can see it is the formation of the archegonia is present in this particular position. So how this archegonial development will take place? Firstly, this is the archegonial initial. Now there will be a single division in which the primary cover cell and the central cell will be produced. The primary cover cell will form the neck initials. Now the neck initials will be staying there to further form the neck cells. The central cell will further divide to form the primary neck canal cell and the primary venter cell. This primary venter cell will further divide to form the ventral canal cell and the egg. So the neck cells will be present at the top, then there will be this ventral canal cell and at the bottom there will be the egg cell. So here it is the structure of an mature archegonium where we can see that this archegonium is a short flux-shaped structure and it is embedded inside the gametophytic tissue. Only the upper tier, that is comprising the 4 neck cells, will be exposed. This neck cells is present in 2 tiers of 2 cells each. After the complete formation of the male gamete and the female gamete, fertilization will take place. Water is very much necessary for fertilization. As I have mentioned, the anthrozoids are provided with flagella and they are motile. So with the help of the water, they will move towards the egg and through the neck of the archegonium, it will reach the egg. There will be a nuclear fusion between the nucleus of the anthrozoids and the egg and ultimately a zygote will be formed which will be diploid in nature. So with this, there is the end of the gametophytic generation and it will initiate the formation of the sporophytic generation. So here we will be talking about the development of the embryo. After the zygote formation, there will be a division in which the epibasal and the hypobasal cell will be formed. So the hypobasal cell will give rise to the suspension initial and the epibasal will form the embryo initial. So there will be this further development. You can see that the suspensor will grow longer. At the bottom there will be the stem initial. At the top the cotyledon will be formed. There will be much differentiation. The proper stem will be formed. And here you can see that the cotyledons have been grown to a much height, so as the stem. So in this mature structure, what we can see, a suspensor cell, the cotyledons, the ligule is slowly getting formed, the stem portion, the foot, and also the rhizophore initiation has also taken place. So on an ending note, we can say that the whole thing that we discussed comprises of the life cycle of Selaginella. So to start with is the sporophytic plant body. The leaves is spirally arranged in the stem, and we can see that there is this strobulous portion. So if we get into the TS of this strobulous portion, we will see that there is this microsporangium and the megasporangium, followed by the microsporophyll and megasporophyll. This microsporangium producing the microspores and the megasporangium producing the megaspores. These megaspores will finally develop into a structure that is a female gametophyte, which will have the archegonia, and the microspore will develop into the male gametophyte that will form the sperm. Now the sperm and the archegonia, that will form the egg, that will have the egg rather, will get fertilized in presence of water. With this, the haploid stage will get ended. Now the zygote will be formed, which will be 2N, and from the zygote further development will take place, and ultimately a proper sporophytic plant will be formed.